Okay, so now here's again the kind of thing where they clarify some of their benchmarks. That's like what they want you to know. This is EOC stuff again. It's in red. So structure related to function, I already pointed that out. Compare and contrast plant and animal cell structures, right? That's what we're getting to more of. We've already had a little bit of that, like cholesterol, right? And then compare and contrast prokaryotic and eukaryotic cells we already did. So here now we're going to focus on these eukaryotic cells that are plant and animal cells and all their different parts. So here is a little thing, and I'm gonna correct some of this a little bit here. There's some common mistakes that are made in textbooks. They're oversimplifications. I won't say mistake, but they're oversimplifications. But this is true in animal cells. You're not going to find these things. All right. We'll come to all those things except maybe that one. And then on this slide, in plant cells, you're not going to find, well, actually, in plant cells, about the only one of these you're not really going to find is this. This is the exceptions I talked about. You'll find these in some plant cells, but they're kind of rare. And so we're gonna pretend that it's a difference, and we're not gonna fret over it. Okay, so part number one, that if you want to, call a part of a cell more important than another part, because they're all important, but you'd probably pick the nucleus to be the most important part of a cell. That one that again makes these eukaryotic cells. The structure of a nucleus is that its membrane is actually a double membrane. 2 phospholipid bilayers, right, is what that word double means. And where the 2 membranes are fused, there's a big hole that allows, as you would logically expect, big molecules to pass in and out of the nucleus. Here you see a diagram of some of those pores. Here you see actual micrograph of some of these pores. They're made by proteins kind of forming a ring, and they let big molecules in and out of the nucleus. Okay, what's in the nucleus? Well, as we talked about the word cytoplasm and protoplasm, you might realize this is a good word for everything that's in the nucleus. Nucleoplasm, okay? That includes DNA and some proteins and a lot of water and a lot of ions and things like that, right? But we focus almost exclusively on the DNA inside the nucleus, and that long stringy, fibrous is stringy, stuff made of the chemical DNA and some protein stuck to it is called chromatin when it's all spread out. But the word we're more familiar with, chromosome, actually only forms when a cell's getting ready to divide, which we'll study again later in a different unit. And all those real thin, stringy chromatin fibers coil up into those little bodies that we call chromosomes, which you and I have 46 of in each one of our cells. Dogs have 98, fruit flies 8. Every kind of species has a distinct number, right? But not a unique number. If you're a plum, you also have 46 chromosomes in each cell. Okay, so this real fast, because this is going to be like a whole unit, and really part of the whole rest of the year we'll refer to this thing. The nucleus is the control center of the cell. This you could have told me when you walked in the door this year, right? That's a common term that you learned in middle school, and you might have learned this, you might have learned more, but control center of the cell. Well, that's kind of what the brain is for your body. You can make that analogy, but the nucleus doesn't control things like your brain does, making conscious kinds of decisions and things, right? What happens in a cell that needs to be controlled? We've said this all year so far. What is life? Chemistry, but very specific, well-controlled chemistry. So that's what needs to be controlled. What do you find in the nucleus? This stuff made of DNA. Chromosomes. What does it do, these chromosomes? I'm going to make this analogy for the first time. You might have had this before, depends on what you've had, right? Chromosome, gene, DNA. You kind of know those terms are all related. How are they related? Because they're not the same. I'm going to make an analogy that's a really good one, and it's commonly made, and we're going to make it a lot later too. A chromosome is like a cookbook. A cookbook has a whole bunch of recipes in it. A gene is like one of those recipes. On a typical one of your 46 human chromosomes, you've got about 1,000 recipes, about 1,000 genes. A gene is a section of that chromosome, just like a recipe is as an actual physical section of a recipe book, right? What does the recipe do? It's the instructions to make something. That's what a gene is. What's a gene? The instructions to make a protein. What do proteins have to do with chemical reactions? Those kind we call enzymes speed them up. And so, because what this control means is to have the right chemistry happening at the right time, and the nucleus has all the recipes to make all the different kinds of enzymes, to make the right reaction happen at the right time, the nucleus is going to release the recipe to make the right enzyme at the right time. And how it does that and all that we'll talk about much later, or actually second quarter, when we focus more on how DNA does that. For right now, that's as far as we're going to go with the function of the nucleus, much more specific, at least a little more specifically than it's the control center of the cell. We want to up our game a little bit. Okay, so we said that ribosomes help build proteins. If you want to make the analogy, they're kind of like the chef. They're going to take stuff from the nucleus and they're going to use it as a recipe to make a protein. Okay, they themselves are made of protein and that other kind of nucleic acid called RNA. And what they help carry out is, again, protein synthesis. Some of the ribosomes are found stuck to membrane that's called the endoplasmic reticulum. The endoplasmic reticulum is made of membrane and it's all throughout a cell. Accounts for half the membranes of a eukaryotic cell, right? It's continuous with the nuclear membrane, and it's actually like a bunch of bags and tubes that have an inside and an outside, as we'll see. So here's some diagram, right? You can see in a couple places here, especially if you enlarge this, that the outer nuclear membrane is actually continuous with this endoplasmic reticulum membrane. You can also see a little bit that there's little dots on this, making it look rough under the microscope, which is why early microscopists simply called this rough ER. It's got ribosomes stuck to it. But then there's ER over here that's connected to the rough stuff but doesn't have ribosomes. And therefore it looks smooth under the microscope. And so this is simply what these kinds of endoplasmic reticulum came to be called. So the rough endoplasmic reticulum, since it has ribosomes physically stuck to it, that suggests correctly that its function is to be a surface on which proteins can be made. The smooth ER is also a surface. It's got a lot of enzymes in its membrane, but it doesn't have ribosomes. So it can't make proteins on the smooth ER, but it can make other molecules, especially like lipids, especially like the phospholipids that make up membranes. And then the smooth ER also has enzymes that help break down glycogen, remember that, polysaccharide, animal starch, into glucose monomers when your blood sugar gets low. And since glucose is blood sugar, by breaking down glycogen and releasing those glucoses, your blood sugar rises. What chemistry is that? Breakdown? Hydrolysis. Good. So this is probably common sense, but it's also a pretty common test kind of question. If you've identified that rough ER is a protein maker, right? And you're told a story that these kind of cells specialize in making proteins, then it stands to reason that you're going to find a lot of rough ER in those kind of cells. Compared to like a fat cell that's not very active, that just kind of stores fat, where you'd expect to find less rough ER, and you will find less rough ER. Okay, so now we're going to look at the process that involves this rough ER and a few other cell parts, and with a goal in mind, so to speak, of making proteins and then secreting them. This takes the combined effects of several cell parts, and it's a very common kind of question backbone to ask you questions about a lot of different cell parts. So here we see a word vesicle. Vesicle is just a little membrane bubble. By membrane bubble, we mean it's made of phospholipid bilayer membrane, and it's a bubble that's got stuff inside. And its function is either to keep it stored in there for later use, or maybe even to help it get carried outside. So a player in this story is the Golgi apparatus. Golgi is always capitalized because it's a guy's name. Sometimes this is called the Golgi bodies, sometimes just the Golgi, so you might see all those different ways. It's all talking about the same thing. Maybe in middle school you learned the Golgi is like the post office of a cell, where things are packaged and sorted and shipped out. That's an okay analogy, but like, you don't want to write that on a test if you're describing Golgi. You want to say what it actually does, and if you use the analogy, make sure you explain the analogy. Okay, so this again plays a role in sending stuff out of a cell, packaging and sending. And so for those cells like the pancreas one that are specialized for secreting things, the Golgi plays a big role, like we'll see. Then you have those cells called lysosomes. Lysosomes are vesicles, but they have their own specific name because somebody gave them their own specific name. They're a vesicle, membrane sac and vesicle are like the same thing, of enzymes that are hydrolytic. They're going to break down polymers into monomers inside of a cell. And that might be for a couple different reasons, as we'll get to. So here we go with all these players we've talked about, or most of them that we've talked about. Here's the rough ER. Proteins are made in there, packaged up in a little vesicle, which actually pinches off of the ER, like you'll see in an animation, and takes that to the Golgi. They made the Golgi a different color here, but it's really the same as the endoplasmic reticulum. It's flattened bags of membrane. And this transport vesicle now is going to merge with the Golgi, and now those proteins are inside the Golgi, these flattened bags. They get often modified a little bit. That means some chemistry is happening. And then once they're kind of finalized, they get pinched off in a little vesicle. And in this little diagram here, they're showing the formation of those vesicles we call lysosomes. Lys, do you remember that? Hydrolysis, lys means split. Well, here it also means split. These are bodies that help split things with their enzymes that are inside. What kind of things would get split? Well, some of your cells, like white blood cells, take in big macromolecules, sometimes even entire things like bacteria cells and viruses, if you're a white blood cell, and they're taking them in to eat. But just like your lunch, you have to break down those big molecules first. So this vacuole or vesicle, vacuoles are just big vesicles, we'll get to those in a second, right? That's got food in it, a food vacuole, is going to have the enzymes of a lysosome emptied into it and break that up into little molecules that then come out and do what they need to do in a cell. Or sometimes cell parts, like this is like a part of a mitochondria, it doesn't work anymore. Something went wrong with it. But it's made of good molecules. And so they're going to get recycled with the help of a lysosome breaking them down. They're going to get recycled so this cell can use them again. So one of the other things that lysosomes can do is to digest enough molecules in the cell to destroy the cell. This so-called programmed cell death, or word's not up here, so I'll write it. Apoptosis is another word for that. This is a very key process for all different kinds of reasons in life. We talk about those more in AP biology, a lot of them. But here's an example of one of them. When you're a little kind of thing, we would call you an embryo at this point. And when I say little, I mean you're like a half an inch long, right? You start to have little arms and legs. But your hands are kind of like paddles. You don't have fingers formed yet. And your fingers don't grow out of those paddles like theoretically they could. What happens is the skin in between what's going to end up being your fingers, that skin, those cells, their lysosomes open up and those cells die at the right time, programmed. Cell death is almost always a good thing. It's a natural thing. It's a normal part of, in this case, your development. But that's how your fingers form. That's kind of neat. Okay, so continuing to go through our cell parts, we have vacuoles. We just mentioned that they're just membrane bags also, just a little bigger. Two different words we really could have done with one. Vacuoles are bigger than vesicles. So we had the food vacuole we talked about. We have a kind of vacuole that can contract with the help of some other cell parts. We'll see some of those in a little animation in a little bit. And they can pump excess water out of a little kind of one-celled freshwater thing called a protist. And then we have one that you should remember, a specific kind of vacuole, simply called a central vacuole, not necessarily because it's in the middle, but that's its name. And this is a plant cell, animal cell difference. Animal cells might have what you call vacuoles, especially like a food vacuole in one of your white blood cells, like we just talked about. But only plant cells are going to have a very, very big, single, very big vacuole called a central vacuole. So there is one. This is a picture micrograph. This is a diagram to show that central vacuole, and it can often take up even more space than this. Not every plant cell will have one of these, but only plant cells will have these. And a lot of plant cells will have them. It's not a rare thing. So this picture right here really should be all you need to see to say, oh, that's got to be a plant cell. And then chloroplasts, which we're getting to, obviously identified as a plant cell. And it helps that they're labeled because sometimes they're hard to just know what that is from a micrograph.